Welcome back to Antibody Basics by Biointron Part 6. In this episode, we'll talk about antibody-drug conjugates, ADCs, the drug type taking the biopharmaceutical industry by storm. What are antibody-drug conjugates? They are composed of a monoclonal antibody covalently bonded to a biologically active cytotoxic drug via a chemical linker. The monoclonal antibody can then bind to a specific target protein or receptor, allowing the delivery of the drug into the target cell. This combination is valuable in cancer as it allows for a highly specific targeting and potent killing effect. The ADC revolution began in 1900 from Paul Ehrlich's magic bullet concept of targeted chemotherapy. His magic bullet would target a cytotoxin to intended structures in unwanted cells, but spare healthy tissues. In the 1980s, the inception of hybridoma technology for generation of monoclonal antibodies helped realize Ehrlich's vision with the first ADC trials underway. In 2000, the first ADC was produced, gemtuzumab ozogamicin, a CD33 antibody conjugated to an anti-tumor antibiotic, calicheamicin. In the present day, we now have a growing understanding of the ADC mechanism of action, and technological breakthroughs have heralded the approvals of over a dozen ADCs. ADCs have several advantages over traditional antibodies. The main advantage is targeted delivery. The combination of the unique targeting capabilities of monoclonal antibodies with the cancer-killing ability of cytotoxic drugs increases the cell-killing potential of traditional monoclonal antibodies. ADCs can also achieve accurate and efficient elimination of target cells without harming the healthy ones. This discrimination between cancerous and normal tissue is advantageous over traditional chemotherapeutic agents. ADCs also have biodegradable linkers which can be either cleavable or non-cleavable. This linker design plays a critical role in modulating stability in the systemic circulation and payload release efficiency in the tumors, which thus affects the ADC pharmacokinetic efficacy and toxicity profiles. An ADC has a tripartite structure: the antibody moiety, linker, and a cytotoxic payload. The antibody component determines the ADC's duration in the bloodstream, immunogenicity, immune functions, and target specificity. Most antibody-drug conjugates use IgG1 as it offers advantages like long serum half-life and strong Fc-mediated immune functions, including ADCC activity. To mitigate potential immunogenic reactions, murine antibodies have largely been replaced with chimeric or humanized ones. An ideal target should be expressed at high levels on the surface of tumor cells and not onto normal cells. Examples of this include HER2 and TROP2. Linkers make sure the payload is bound to the antibody while in circulation and later released at the tumor site. There are two types. The first, cleavable linkers, releases the payload upon reduction, proteolysis, or hydrolysis. Non-cleavable linkers require complete lysosomal degradation for payload release. Non-cleavable linkers provide stability to the ADC during circulation and may contribute to a better safety profile, but may also hinder the potential for bystander killing. This is when the payload is released into the tumor microenvironment and kills antigen-less cancer cells or cancer-supporting cells. Payloads are the chemotherapeutic agents which exert cytotoxic effects on the tumor cells targeted by the ADCs. Usually, they are microtubule binding or DNA-damaging inducing agents like calicheamicins, auristatins, and maytansinoids. Newer payloads include topoisomerase I inhibitors like camptothecin derivatives. Further development of more powerful ADCs can have varied payloads like immune-stimulating agents which are non-chemotherapeutic. Progress is also being made in improving the therapeutic window and decreasing systemic side effects to healthy cells. Currently, all 13 approved ADCs are focused on cancer. Here are some examples: The first ADC to be approved in the world is Mylotarg. Approved in 2000, it targets CD33 to treat acute myeloid leukemia. Another example is Kadcyla, which was approved in 2013 and targets HER2 to treat early or metastatic platinum-resistant breast cancer. It is the top-selling ADC with sales revenues of about $2.2 billion in 2022. Another ADC is Trodelvy, which was approved in 2020 and targets TROP2 to treat metastatic triple-negative breast cancer and metastatic urothelial cancer. And finally, we have Elahere, which was the most recently approved ADC in 2022. It targets FR-alpha to treat fallopian tube cancer or primary peritoneal cancer. As we look towards the future, third-generation antibody-drug conjugates are in development. Up until 2019, all ADCs on the market were heterogeneous mixtures, meaning there were variable numbers of cytotoxic drugs on the monoclonal antibody in various places. To broaden the therapeutic index of ADCs, regio-specific versions were introduced. There are three main methods for this: bioconjugation onto natural or non-natural amino acids, bioconjugation using enzymes, and linker-based bioconjugation. An alternative ADC format may be smaller formats. Most ADCs targeting solid tumors have failed, likely due to the large IgG structure. Therefore, smaller formats like peptides, single-domain antibody fragments, single-chain variable fragments, antigen-binding fragments, and small immunoproteins hold valuable potential. Another development may involve new targets. Since targeting antigens on the surface of cancer cells can be difficult in solid tumors rich in intercellular stroma, we may instead target the tumor microenvironment, stroma or vasculature. Thus, extracellular proteases and other matrix components could be used for extracellular release of cytotoxics. Furthermore, we could target cancer cells with low antigen expression or resistance to auristatins or maytansinoids with PBD dimers. They are about 50 to 100 times more effective than conventional cytotoxics used in ADCs, such as MMAE or DM1, demonstrating picomolar activity against several human tumor cell lines. Biointron focuses on antibody discovery, expression, and optimization services. Our main feature of antibody expression allows your gene sequence to become purified antibodies in only 2 weeks. Our high-throughput platform enables the rapid expression of recombinant antibodies in HEK293 or CHO cells. We guarantee the delivery of 100 micrograms to 100 grams of recombinant antibodies. And no antibody means no cost. Biointron is also a well-recognized leader in VHH antibody discovery and production. With our self-owned alpaca breeding farm, we offer a 1 alpaca per 1 project commitment and guarantee the delivery of over 20 unique binders, high diversity, and large capacity. We also have several other services, including but not limited to bispecific antibody production, afucosylated antibodies, single B-cell screening, hybridoma sequencing, antibody optimization, recombinant protein expression in the mammalian system, and CHOK1 stable cell line generation. Thank you for watching, and comment with any questions or topics you'd like us to cover next. Don't forget to like and subscribe for more.